Okay, we're going to get started. So it's really a great pleasure and honor to be here today. So my name is Michael Schatz. I'm one of the program directors for the AnVIL project. I'm also a professor at Johns Hopkins. It's really a great honor to be here. I think this meeting is, I don't know, something like 7 or 8 years in the making, to be able to all come together to kind of celebrate and just discuss and just get excited about AnVIL and everything we've done so far and talk about some of the ongoing activities about in the future. Before diving into the main program, as I was sort of talking to people this morning and last night, it occurred to me that many people here have never been to Cold Spring Harbor before, and I just wanted to say a few words about this campus and a little bit about how we came to be here specifically here today. So Cold Spring Harbor is one of the oldest research institutions in the United States, going back to the 1890s. In the early days, there was a marine research station going out in the harbor here. In the early days, there was a big agricultural component looking at sort of major crops. In the early 1900s, there was a big major focus in human genetics and try to lay down some of the fundamental principles here. It's a really beautiful campus. I think we can all agree. Just it's like living inside of a postcard. I was on the faculty here from 2010 to 2016, so I feel like it's always a homecoming when I come back here. So this part of the campus here is called the lower campus, and there's facilities for conferences and meetings. There's something like 10,000 visitors a year that come to this campus. So a lot of people have had some experience. I really strongly encourage you, if you get a chance at one of the breaks or maybe in the morning, take a walk on campus. It's a beautiful park. If you go down towards the beach, that's actually where I got married, on the water there. It's just a spectacular campus to be at. So Cold Spring Harbor has this enormous history about really fundamental discoveries in molecular biology. So here are just a few of the Nobel laureates that have passed through here, really making really fundamental discoveries into kind of the origins of life. So this includes Barbara McClintock, who discovered transposable elements, so-called jumping genes, in studies in corn. We had Alfred Hershey, who designed and executed some of the first experiments that demonstrated that DNA was the molecule that transmitted genetic information as opposed to proteins or other information. We had Rich Roberts, who designed and executed some of the first experiments to recognize splicing was a major feature of RNA. And Carroll discovered and worked out the mechanisms for DNA replication, especially at the ends of DNA chromosomes in the telomeres and the telomerase enzymes there. So there's just been a lot of really fundamental discoveries about life that have been made right here on this campus. It makes it a really special place to celebrate anything sort of related to molecular biology. Cold Spring Harbor also has a very strong tradition in quantitative research. The first symposium of quantitative biology was held in 1933, almost coming up on 100 years ago. And every year since, there's been a symposium kind of talking about different mechanisms, about different characteristics, different quantitative aspects that have been going on. So there's a very long tradition of that. It's a really fundamental part of the biology and the science that has taken place here. In addition to kind of the scientists here, this has been home to many outstanding visiting scientists in quantitative biology, molecular biology, and beyond. So this is a photo. Does everyone know? This is Claude Shannon, who invented something called information theory. If you've ever heard of Shannon entropy, basically this is like how all information in the world is transmitted, is based on some of the fundamental principles that Claude Shannon sort of laid out. So Claude Shannon completed his PhD at MIT in electrical engineering. That's well known, and then went on to Bell Labs to kind of do his research. What you might not know, though, his PhD was actually on an algebra for theoretical genetics. And the work he did for that work was here at Cold Spring Harbor. He came and kind of learned and got inspired by just sort of the patterns of genetics and the patterns of life around him. So there's a really long, old tradition about these really fundamental quantitative discoveries and theories being developed right here. So it makes a really special home for this. Another kind of milestone for many of us was the first Galaxy conference was held here at Cold Spring Harbor back in 2010. It was kind of a day-and-a-half meeting. In fact, this meeting, the AnVIL Community Conference, is very much inspired by that original Galaxy conference. One of the big headlines, and I think Ennis is here and maybe can comment on it, was talking about how to run Galaxy in the cloud for the very first time with this very exciting UI. I found this slide deck, and it talks about, go into the EC2 console, boot up a virtual machine, deploy the dependencies. So in some ways, we've come a long way, and in some ways, I think we're still right where we started some 14 years ago. Back in 2010, it was, like as I mentioned, was also when I joined the faculty here. Another kind of important milestone was there's many conferences that take place here. I was one of the co-organizers for a conference called Genome Informatics with Jennifer Harrow and James Taylor, our dear friend who passed away a few years ago. We were co-organizers of a conference called Genome Informatics. This is our Halloween conference, hence the kind of silly cover here. And it occurred to us, well, genome informatics was emerging and there was a lot of excitement around it and a lot going on, but that's only kind of like one sliver of sort of modern molecular biology, right? There's beyond sort of genomics, there's other kind of molecular data we want to collect. There's imaging data we want to collect. There's EHR data we want to collect. There's phenotypic data we want to collect. So genomes, of course, are very special and near and dear to us, but there's more in the space that we really wanted to celebrate. And that's what really inspired the formation of sort of a sister conference, Biological Data Sciences. This is our 10-year anniversary of that meeting that actually starts immediately after this conference. So why are we here at Cold Spring Harbor? Why on these days? It's because we wanted to be, for our kickoff, we wanted to be kind of partnered up with Biological Data Sciences in the same way that the original Galaxy meeting was partnered up with Biology of Genomes. So many of us will be at the main conference, starting Wednesday night. That's great. Some of us cannot stay. That's great too. But I think it'll be a sort of a nice sort of companion meeting to the kickoff conference that we're having here today. So we have a really exciting agenda for us today over the next 2 days, just to kind of quickly review. So this morning we have kind of a few introductions, talk a little bit about the past, present, and future of AnVIL, about where we came from, some of our current activities, give you a little bit sneak preview into the future, some of the things that are kind of in the works and coming soon. After some presentations internally, we've invited a few really outstanding speakers to come and talk about their experiences with AnVIL and sort of cloud computing more generally. So our first keynote speaker right here, Tyshell Turner, talking about her work in neurological disorders and using AnVIL to power some very large analysis. I think I just saw, how many genomes was it? 30,000 genomes, kind of crushing through using the platform. After that, we'll have Anshul Kanjale give a presentation talking about some of his really exciting work in machine learning. Unfortunately, he got super sick just the other day, so he's going to zoom in for that. But then Vivek, one of his lab members, will be here to kind of run the CoFest after that. And then tomorrow morning, Benedict Payton, one of our friends and colleagues, is going to be talking, I think, about some clinical work and some pan-genome work, talking about some of the new resources that are available. And I think a lot of that will be exciting work powered by AnVIL. So in addition to those sort of, kind of presentations and sort of broadcasting of information, we really want this to be a dialogue. We really want to kind of, hear from you all, what's working. Also, what are places where we need further work for optimization. So there's a lot of time sort of built into the schedule to get some hands-on sessions, our so-called CoFest, where you can kind of sort of organize into a few sessions related to everything from just getting an introduction to the platform to some more advanced topics of machine learning, polygenic risk scores, and then also kind of an open forum just to kind of discuss more technical components of the platform. So you're invited to attend those breakout sessions. And then tomorrow, we're also going to have what we're calling just a town hall. So this is a very sort of open-ended discussion about what is working, what's needed, some of the maybe larger trends beyond NHGRI, some of the activities that are present there. So I think that'll be a real lively discussion about sort of the future of genomics research, biomedical research, and beyond. So one thing that we wanted to do up front was, we had, prior to the meeting, we did send out a survey asking for your impressions about which breakout sessions were of interest to you. So what we like to do is invite you to kind of take an updated version of the survey so we get a sort of a sense of what people are interested in. Also, during the town hall, we have seeded it with a few discussion points, but if there are any specific topics that you really wanted to bring up, that would be a great time to kind of just sort of make a quick note. We don't need a novel or anything, just a quick note about what sorts of things you're interested to discuss during the town hall, so we're well prepared so that we can kind of really sort of match your interests here. So there's a QR code. You can also go to bit.ly/ACC, that's AnVIL Community Conference 24-breakout. We'll get you just to this a really short, brief Google form to kind of collect that information if there's topics that you'd like to talk about. So that's sort of the overview. So I thought now we could kind of dive more into the program and talk more about where we're coming from. So as I said, I'm one of the co-directors of the AnVIL platform. I'm joined up front here with Jonathan Lawson from the Broad. So his team at the Broad it manages a major component called Terra. That's one of our key components for kind of organizing the whole platform, where data are organized, where various analyses are launched from. I'm also joined up front by Robert Carroll from Vanderbilt University. And really, they have a focused mission there to improve clinical genomics research and support those activities through something called the ACR. But really, we're just the tip of the iceberg. Behind us are dozens, maybe hundreds of individuals working across dozens of institutions that are all coming together to support this one amazing platform to kind of empower next generation of science and genomics research. So there's a few of us up front, but we're really just here on behalf of this much larger team. I just really wanted to thank all of you for all your contributions to this outstanding work. So just to kind of put a little bit of context about where we're coming from and why we're here, right? So I think we can all recognize in the field, there's been a tremendous growth in terms of the amount of data that are being generated and sort of the sophistication of the analysis that needs to take place. These days, it's quite common to have a single research project at the scale of like tens of thousands of genomes, hundreds of thousands of genomes. Within the United States, the largest project that I'm aware of is the All of Us project, which is on track to sequence the genomes of 1 million US citizens and be able to do all kinds of analysis about associations between health and disease through that platform. So there's this huge growth in the amount of data being generated, the types of analysis that needs to take place, and a lot of the research is just becoming really sophisticated. So in the early days when I got started in genomics, I don't know, more than 20 years ago, kind of step one would be to download one of these ginormous data sets and try to do analysis locally. And today, that still exists to some degree, but for certain projects, that just becomes incredibly difficult and impractical to do so. So instead of bringing all the data to the researcher, wouldn't it be easier, wouldn't it be more streamlined if we could have a single platform where researchers anywhere in a very democratized way could access the same data, access the same analysis tools, get access to advanced hardware, advanced software, computing, have access to that great infrastructure that could be shared. So there's a lot of sort of upside to this about reduced redundancy, reduced costs. We can have kind of one secure perimeter that can kind of monitor anything, and then everyone can work inside of there. We can have greater accessibility where it's just not very privileged people, but anyone, anywhere can access to the cloud. The cloud is all about democratization of science. And then another major component is that elastic nature, where if you just need a little bit of compute, no problem. We can give you a little bit. If you need a lot of compute, no problem. We can sort of scale up and scale down as your requirements for your project need. So AnVIL is this cloud orchestration system where we're going to be able to work with data that comes from NHGRI that are kind of available within the AnVIL. We're also going to be able to work with other cloud resources through something called NCPI, the NIH Cloud Platform Interoperability Project, where we can work with other institutes around NIH. We can also bring in data from your own institution. And we want to make sure there's great support for that. Bringing all these data, bringing all these analysis tools together inside of one platform where all this research can take place. So what is AnVIL? In the same way that on a laptop, there's, I don't know, a web browser, email client, calendar, maybe a few games, maybe a few other things. But they all sort of work together through a collection of kind of APIs, file systems, sort of more technical components. So you get many applications all running in the same system. AnVIL is organized in a similar way, where there's many sort of applications, but they all work together, all tied together to be able to have one sort of environment where you can do analysis. At the top of the list is that Terra platform that I mentioned before. This is our key launch pad for what we call workspaces. This is where data get organized, where analysis tools get launched from, where interactive analysis, big, large batch workflows as well can be launched from. Other components include something called the Terra data repo, where data can be indexed, metadata can be indexed. Something that we're really striving to do is this ability to generate what are called synthetic cohorts. Now that we have hundreds of thousands of genomes already loaded into AnVIL, instead of going and resequencing a new population, maybe we can go through an existing data set to find those individuals that have the characteristics that we're interested in. So that's sort of the type of queries that we want to be able to support those, identify those individuals, bring them into a new workspace where you can do analysis of them. Another major component is DocStore. This is our platform for sharing analysis workflows and tools, notebooks, which is a really great way to have reproducibility, interoperability, where now one analyst can develop their own approach, share it with the rest of the world, where others can learn from that, build on that, and sort of take advantage of those resources to do additional analyses. In addition to those components, we have several interfaces to do a variety of different types of analysis. Things that'll be very familiar to this audience, like Jupyter Notebooks or RStudio and Bioconductor are available. We also have a full-featured version of Galaxy where you can sort of spin up your own interface and be able to do very sophisticated analysis all inside of a graphical user interface. So one thing that we take very seriously inside of AnVIL is the security of the data, right? These are very personal, very important, very private, precious resources. If someone steals your credit card number, I can issue you a new credit card number. But if someone steals your genome, we're very limited in what we can do. So we take security very, very seriously. In the sort of in the government speak, we're a FedRAMP certified system, meaning that we're sort of authorized to host protected data sets. We also behind the scenes have a lot of encryption, authentication, logging, auditing, and so forth to make sure that the data are very well protected. So AnVIL is a software stack where we have all these applications. It runs in the commercial cloud. Initially, Google Cloud Platform was our main platform for this, but we're also migrating into Azure and multi-cloud as we'll hear about later throughout this morning. One really important notification from NIH is that we are an NIH designated data repository. So if you're back at your home institution, you get questions about what is AnVIL and is it really secure for this type of work, you can point them to this document, which really sort of really clearly just states that AnVIL is the primary NIH designated repository for NHGRI funded data, metadata, and associated documentation. So again, if you ever get questions about that, you can point to that, come contact any of us. We'll help you out, make sure that people are aware of what's available there through AnVIL. So to date, we've collected more than 6 petabytes of data. Something like 600,000 human genome data sets have been ingested and released. I think there's a similar amount of data that is in the queue to be released. So there's just, we have this ever-growing footprint of data that is being sort of organized inside of AnVIL. A lot of this data comes from major consortiums funded by NHGRI. This includes things like the Centers for Common Disease Genomics, the Centers for Mammalian Disease Genomics, GTEx, 1000 Genomes, eMERGE, T2T, HPRC, and beyond. We're also the primary platform for most of NHGRI's current genomics consortium. These include clinical research programs like Regular Primed, kind of more basic sciences ones like IGVF, that are looking to understand how variation impacts a variety of molecular outcomes, and many, many others. So it's the platform, it's the home for kind of all the largest genomics data initiatives across NHGRI. So as I mentioned, there's many entry points into the platform, depending on your background, depending on your interests, depending on what you're trying to do. I had kind of a lively conversation last night about Jupyter Notebooks. I hope you don't get me wrong. I do love Jupyter Notebooks. They certainly have a time and a place, and we love them, we use them. But sometimes you need to be able to do more than that, and that's why we offer kind of multiple interfaces. So I think Jupyter and our studio will be kind of quite familiar here, right? So this is a way where you can do a variety of coding and generate plots and statistics and machine learning. Especially in R and Python, it's very rich environments to do a data analysis. Basically, every paper I write kind of starts with a bunch of analysis, and then eventually we'll get into one of these systems so that we can make those figures and plots and those statistics to be able to do the analysis. So don't get me wrong, I'm a true believer. Another major component is that Galaxy platform. I just wanted to have one slide about what's available there. So this is long-running platform for accessible, reproducible, and collaborative biomedical analyses. So Galaxy has a long history that predates AnVIL by, more than a decade. We just reached more than 500,000 registered users around the world. We host more than 100,000 tools. We've run hundreds of millions of jobs, hundreds of millions of data sets. A lot of the success of Galaxy has come from our extensive training network, where we have more than 400 tutorials available. And very recently, we just hit more than 20,000 citations to the platform. So now inside of AnVIL, you can get your own private, secure version of Galaxy, where you can work with human protected data sets, do analysis in this very accessible and reproducible way. And then the final component I wanted to just sort of highlight for this intro here is something called the Workflow Description Language, or WDL. And this is one of our key technologies for being able to run very large workflows. So if you're doing analysis over thousands and tens of thousands, hundred thousands of genomes, this is probably what you're going to want to use right now. It's extremely scalable, extremely flexible. It is a bit sort of technically demanding in that it's all kind of command line driven, although we will have a session. Allie's here someplace. Yeah. Well, if you're kind of new to AnVIL, new to WDL, she'll get you up to speed about how you can run your own tools. It's very, very flexible. I teach it to my undergraduate students at Hopkins to be able to empower them to be able to do very large scale reproducible research. So just one slide about this. So what is a WDL? What's some of the secret sauce inside of AnVIL? At the end of the day, it's just command line execution. I tease, it's a fancy bash script where you have a command block. Like here we're running samtools stats, which is just a common way to get some information about alignments. And we're going to decorate it with what are the inputs to the program you want to run. You're going to decorate it with what are the outputs. But because we're running in the cloud, we get a lot of flexibility about the resources that we're going to bring to the table. You get to pick how much local disk, how much RAM do we have, how many CPUs do we have, if you need GPU access. this is what's sort of amazing about the cloud is that you can dial in whatever kind of compute resources you need and be able to kind of scale up, scale down as you need. So this is a very simple whittle, just run SAMTools stats, but often these get chained together into much larger workflows that can do very sophisticated analysis at very large scales. So just to give you a little taste about what's been possible. So here's a nice plot from the Centers of Common Disease Genomics project where they've sequenced and been jointly analyzing almost 140,000 whole genome data sets. This is a PCA plot of the genotypes. Normally this would be colored by the ethnicity of the donor, but here it's colored by the sequencing center, where we're getting tens of thousands of samples from Baylor, from Broad, from the Genome Center, from WashU, really trying to make the point where this is a place where very large scale joint analysis can take place. If without the AnVIL, there would be, I don't know, some awkward process where different subsets of the data would be downloaded and exchanged. There'd just be a lot of inefficiencies to this, but now we can do this all in the cloud at very large scales in a very sort of collaborative way. Other sort of major success stories include, working with some of the new reference genomes and some of the really state-of-the-art technologies. So the AnVIL has been the platform for the telomere to telomere project. It has been the platform for the Human Pangenome Research Consortium to be able to do, very excellent genome assemblies. And then once we have these assemblies of new reference genomes, how does that improve our understanding of human genetic diversity? How does that understand of clinical genetics as well? Through large-scale reanalysis of different data sets in the AnVIL using these new reference genomes, all been powered by the AnVIL platform. I think for a lot of people, what gets them excited is a plot like this. So this is a picture of one of my former students, Samantha Zartar, in partnership with the Telomere to Telomere Consortium, where we're doing this massive realignment of many petabytes of data. At Hopkins, it would have taken us the better part of a year, but then we could scale up to more than 13,000 cores. A single grad student pushing one button can get access to 13,000 cores. Took us from, I don't know, a year of compute down to a few days of compute that we were just empowered to do so for a single student. I think that's a very transformative experience when you're empowered to be able to do an analysis like that. Another example, just to very quickly highlight another opportunity, is we can combine different data sets, different data types in very clever ways. So here we can use data from the Human Pangenome Reference Consortiums, the long-read data sets, to discover what are called structural variants. Here, what we're looking at is an IGV screenshot of a heterozygous deletion in one of the donors. We can take that as our sort of discovery platform, identify those types of variation. Then we can go into other data sets. We can go into GTEx, where we can do genotyping in the DNA data. We can then integrate in the RNA data, and we can look for what are called SVE-QTLs. These are where there's a structural variation that is associated with a major change in gene expression that we can then analyze in a Jupyter notebook using bioconductor resources to detect something like 10,000 new associations between those SVs and EQTLs. I also wanted to comment that AnVIL is not just for the Hopkins and Broads of the world, but we also have a focused program to support researchers at smaller institutions. So we work really closely with something called the Genomic Data Science Community Network. This is a network of about 30 institutions around the United States from that have been historically left out of the genomic revolution. So this includes a variety of historically Black colleges and universities, tribal colleges, community colleges, and Hispanic serving institutions. In fact, just a few weeks ago, we were down in Puerto Rico visiting some of our collaborators that are kind of working together, talking about the latest and greatest research that is possible there. So we've been working very closely with them to support research on the AnVIL, again, to make sure that they have access to the latest technologies and the latest genomics research as well. And kind of more broadly, we have this great outreach program, and many of the individuals are here. And I really am so incredibly grateful to all the hard work you put into the platform. I get, really excited, as you could probably tell, I get really excited about the technology, but technology that no one knows how to use or even knows about is not very helpful. So we have this awesome team, spanning all three of the projects, where we all kind of promote and support each other and support the whole community. That's what we're all about here. So throughout the year, we host a number of events. We're trying to partner with members of those different consortiums to have so-called AnVIL champions to kind of support each other, to build up the community. One thing that we've been planning all along was to have a community workshop where we'd bring together people in person. And that's exactly the origin story for why we're all here today. This is something that we know that we wanted and needed to do. And it's just really, really, really exciting after all these years to be able to come together in person to celebrate this research. So as I mentioned, we have a couple awesome community conference speakers that will be kind of talking about their experience with AnVIL and cloud computing and kind of the state of the art in genomics more broadly. And I think that will be kind of a great anchor point to the research that we're talking about. In addition, we're going to have a variety of co-fests. Again, you get to sort of pick and choose which of these different topics. I think what we're going to do is go around the room and have each of the co-fest leads say just a few words about this. Is that okay? Ellie, do you want to kick us off here? Yeah, because I think there's a few people on Zoom that could benefit from the microphone, or maybe we could pass the mic.
Yeah. Can you hear me?
Yes.
Oh, yeah. Hi, I'm Allie. I work at the Broad Institute on Terra, the Terra platform. I'm going to speak about how to wrap your tool in WDL and run it in AnVIL. So you're thinking, what is that? Well, maybe you have something that you want to run as a pipeline, like a Python script, or more likely a Nextflow script. But Terra AnVIL runs WDLs. So I'm going to work through, I'm going to introduce how to run a WDL, how to find a WDL and run it in AnVIL. And then I'm going to work a simple example of wrapping your tool, no matter what it is, in a WDL and running it. And then I'm hoping that together we'll develop a resource for people. I'm especially wanting to make a plug and play workspace where people could run their Nextflow pipeline. So.
Thank you, Allie. Vivek, you want to say a few words?
Hey, I'm Vivek. I'm a postdoc / research scientist in Anshul Kundaji's lab at Stanford. Yeah, so today we are planning to show you guys like how to train machine learning models on ChIP-seq and accessibility data sets, and also use these machine learning models to do some downstream analysis like scoring variance, looking at sysregulatory code, like those sort of things. Yeah.
Thank you. Matt?
All right, so I'm Matt Lebo. I work at ACR. So we're going to be focusing our session on polygenic risk scores and initially giving a primer on what PRS is and sort of like how to think about it and how to calculate it, and then working through different WDLs of more complexity to be able to show people how to run these analyses on the cloud, work we've been doing with eMERGE and internally with our lab. And at the end, we have time, depending upon how far we get, looking at scripts to visualize the WDLs themselves and potentially building WDLs, or sorry, visualize the PRS themselves and building WDLs or using RStudio to do that. So.
Ava?
Hey everyone, I'm Ava. I'm a member of the AnVIL outreach team, and we are part of the brainstorming feature requests co-fest session. And this is more of a catch-all for you to, have a billing project, work on something that you're interested in, and see if there's things that we could do better or that we could add, or things that you really like. We want to hear your feedback there. Also, if you're new to AnVIL, you haven't used it much and want a little help getting started, I can be there to support that. Thanks.
Thank you. And then last but not least, at the Galaxy conferences, we have this tradition of these so-called birds of a feather, where these are kind of self-organized discussion groups around different topics. So by design, there's no preset agenda for those, but if people want to self-organize and have focused discussions on, I don't know, technologies, data types, the future, AI, you name it, I think there's kind of a lot of opportunities there to kind of have kind of self-organized discussions around that. Okay, so what we're going to do now is we're going to pivot just a little bit. That was hopefully give you a broad overview, because again, I know there's a number of new folks in the room here. Now what we're going to do is talk a little bit about our roadmap, about where we've come from to get started, and then really the point of this session is talk more about the future, about what are some of the activities in the works, some really new results that we wanted to be able to share with all of you here. So I'm going to kick this off, but then I'm going to hand it over to some of my sort of friends and colleagues here to kind of round out this session. So AnVIL kind of officially got started way back in October 2018 with the initial awards to the Broad, to Johns Hopkins, to be able to kind of set up the platform. Even from day one, though, there were many other kind of partner institutions involved in the AnVIL, where we knew that it was going to, it was going to just take a large network of different investigators and researchers to all come together to bring about all of the different components of the AnVIL as it is today. So that was back in 2018. In the first year, I think there was a lot of sort of design discussions, a lot of architecture discussions about exactly how AnVIL would be launched and how all the different components would work together. Kind of one of our main milestones was kind of organizing and releasing Terra as part of AnVIL, and then sort of organizing how other components would fit inside of that Terra model to be able to launch them, to be able to organize them. That was back in 2019. In 2020, we had other major components stand up. We had kind of the first release of DocStore. We had the first releases of the bioconductor package for AnVIL. We had the first version of Galaxy that could be run inside of AnVIL. We had some of our first major outreach events. We had something called the Magic Jamboree, where we looked at some of the genomic data sciences teams at NHGRI and supported them getting onboarded onto AnVIL. We had some of our first major workshops for outreach. We had an event at ASHG talking about running Galaxy and AnVIL. 2020, if you remember, was also a difficult year. We had COVID striking. Very sadly, James Tamler passed away in that year as well. In 2021, we had a few major milestones as well. We published our first sort of flagship paper that was a cover story of cell genomics. We had some other major components like Seeker released inside of AnVIL to be able to look for clinical associations. We started piloting something called Duos that we'll hear a lot about in a few minutes. Those of you that have ever been sort of frustrated with getting into dbGaP, Tyshell, should pay attention to Duos. I think there's some real transformative technology coming along the way there. We also partnered with some groups outside of AnVIL to support them with cloud credits to be able to get onboarded. We also held some major workshops talking about the future directions of AnVIL. In 2022, I would like to say was a real tipping point where we had a mature platform. We could do some really exciting science around this. For example, this is when we were able to support the telomere to telomere consortium, be able to support in that research with that new reference genome. We also had other sort of activities. We partnered with the PharmCat team to release one of their pharmacogenomics tools inside of AnVIL. We had another round of cloud credits, again, to onboard new users. We had a publication from that Genomic Data Science Community Network in genome research talking about how we can support researchers at all kinds of institutions. And then in 2023, we're really in our stride here. We had hosted more than 5 petabytes of data. We had more than 300 workspaces. We were starting to see an uptick in adoption. We're starting to see uptick in sort of the publications that were coming out. We had other consortiums like the HPRC, eMERGE, and CSER, all published sort of major papers in that year talking about the use of AnVIL. So today it's a mature platform. When I think about it, there are kind of 3 major pillars. We have a major focus effort on the data that are there. There's a lot of data that you can get access to right away. It's also a place where if you want to bring your own data, that's a great use of the AnVIL. We also have extensive capabilities for different types of compute, where we can kind of, as we've been talking about, we can look at it, we can do analysis of many different forms. And then a third major focus is on the community. It's all about this. It's all about bringing people together, supporting each other, making sure that we can collaborate, that we can share, that we can learn from each other, support each other to get our work done, kind of help each other make discoveries in the world. Just a quick summary of some of the numbers, just really outstanding research, going from basically 0 just a few years ago into now, thousands of workflows that are available, hundreds of workspaces, thousands and thousands of packages and tools available through all the different components. There's just been sort of massive uptick and massive growth on the platform. One thing I'm particularly proud of is from our flagship paper. Just a few months ago, we reached our first 100 citations to the paper. In fact, as you look around the room, that's what all of those cover pages are. We printed out the first 100 pages. I really encourage you just to kind of walk through. It's just really exciting to see the diversity of the research. Makes it all come alive. So it's been exciting working on AnVIL, but to me, seeing all these papers is what it's all about. It's all about supporting the work of others and being an enabler to incredible science. And it's a dream to see it all come true. And it's just incredibly impactful. So that's our kind of past and present. What we do now is talk a little bit about the future here. So I think one of our, as we mentioned, one of our core pillars has been the data and the data sets that are available. As I mentioned, we're engaged with basically all of the major consortium and NHGRI generating very large data sets. We're also talking to individual researchers with R1s and other types of awards, trying to make them aware of the AnVIL, hopefully encourage them to load their data, work with them to do analysis on the platform. So we really want this to be a platform for everyone. And I think that there's a real opportunity as all these different data sets are being loaded. There's a real opportunity to do harmonization and integration. This is a plot here. By these days, this is actually just a small representation, but this is a plot from the GCHEX consortium. That's the single most popular data set of all of NIH. And I think one of the great hallmarks of it has been how it's been so nicely organized, where you can just download a matrix of gene expression across about 1,000 people, across dozens of tissues. And it's just, it's the premier data set to understand how genetic variations impacts gene expression. And it's all because it was so nicely harmonized and it's so accessible, and it's just an amazing resource. So this is the type of activity that we're looking to more broadly present to the world through all the data sets that we have. And it's just a massive opportunity. In addition to kind of the potential new discoveries that can be made here, I think another kind of benefit to this by having fair data and fair tools is that we can be self-critical and we can point out when maybe there's been some missteps that have taken place. So in the cancer world, there was a major paper that was published in 2020 that was sort of highlighting that there could be an association between the microbiome in different tissues and cancer diagnosis. And then a different set of researchers went back to that same data, took a really critical look at it, and realized that there was a number of problems with the analysis that was taking place there, that the types of signatures that we're finding for the microbiome just didn't make any sense there. So what I'm trying to say here is there's great upside to bringing data together, and also this is a great way to make sure that our results are reproducible and reliable. If we're going to be making clinical decisions based on genomics data, we want to make sure that these are valid results that we can make sense of, and it's so important that the data are accessible to be able to support them. So already in AnVIL, we have a number of components to support so-called fairness. I'm not going to go through all of them one by one, but this is findable, accessible, interoperable, and reusable. These are kind of core principles that we really support and engage with. We're trying to use all of the sort of appropriate standards, technologies, and so forth to make sure that our data, our metadata, our analysis are all fair. As we move forward, there's a number of new services that are being stood up and new resources that are being stood up. I wanted to kind of announce to everyone here. And our goal here, and this comes from kind of one of our proposals, our goal is to make the research environment richer and more powerful by providing these scientific services and pre-computed resources for these critical analysis steps. Basically, our goal is empower you to do everything that you could do locally, but even more efficiently and more effectively in the cloud inside of AnVIL. That's sort of one of our overriding principles as we move forward. And there's kind of 4 major pillars that we wanted to highlight for this. So the first pillar that is sort of emerging is, I'm sure everyone is aware about the AI revolution that we're currently inside of. And we want to make sure that there's really strong support from machine learning services and resources to be able to run kind of popular ML frameworks all inside of AnVIL. So a couple specific initiatives is working with the Terra team and the Galaxy team and bioconductor teams. We have access to things like scikit-learn, to PyTorch, to KRAS. We have Galaxy ML is a sort of point-and-click user accessible way to be able to run these models. We're also in communication with the Kapoy team, which is a model zoo where you can do very sophisticated analysis. And I think that's some of what Vivek's going to be talking about. It's how we can use that and other resources to bring in some of these great models. But I think this is just the very beginning. Just a couple months ago, there was an entire issue of Nature Methods dedicated to advances in AI and biology. And I'm sure you all saw the Nobel Prize Awards just a few weeks ago, many of which were sort of leveraging AI technologies for a variety of biomedical applications. Another sort of major focus is being able for data and metadata harmonization, where there's a huge amount of genomics data that is being ingested. And I think there's an opportunity to learn from that genomics data in order to make the data sets richer. So within our teams, we've had some successes along these lines. There's a major project called Recount, where we go back and reprocess basically all available RNA-seq data in the world, more than 700,000 data sets that have been processed through Recount. We want to make sure resources like that are available so that we can do very deep investigations into gene expression and splicing. I like to think Rich Roberts would be proud of that project. Another major one is phenopredict, where once data sets are loaded, sometimes there's really accurate, really extensive notes and metadata about where the sample came from and the tissue type and the technical characteristics, but not always. But there's an opportunity to use machine learning to be able to go back and make predictions about those phenotypes, make predictions about those metadata, make predictions about those technical components is a way to kind of have harmonized quality control data loaded to be able to empower your research. Another major focus, and this has been a focus of my own research lab for many years now, has been kind of advancing on genomics using long-read sequencing technology. The premier example of this has been the telomere to telomere project, where we're able to get every single nucleotide of a genome actually resolved for the first time. But beyond that single reference genome, there's new opportunities to be able to use these technologies for clinical research, and that's what Benedict's going to be talking about. And one of the activities that we're supporting is a sort of a crowdsource database of variation that can only be discovered using long-read sequencing. So there's something called ColorsDB that we've been working with that is sort of an aggregation of existing data sets that we've been processing in a harmonized way and being able to release the variant data sets with that. At the Biological Data Sciences Conference in 2 days, there'll be a poster about that ColorsDB database where to date we've been able to aggregate something like 2,000 different samples to have our first release of the database. And the idea is if you're sequencing a new clinical sample, you can then screen your variants through this catalog. And if you see, oh, these are actually common variants in other people, they're common variants, they're probably unlikely to be related to a clinical phenotype that you're studying. So we hope that this will help focus in on those clinically actionable variants much more quickly and accessibly. Another kind of opportunity is to try to more broadly do harmonization of our data sets inside of AnVIL. Right? Historically, a lot of the research has been using GRCH38 as our sort of reference genome. It's very well understood, it's well used. There's extensive annotations around it. But as we kind of move into the modern era, we've kind of come to realize that GRCH38 also has some weirdness to it. And there's some errors in it and some gaps in it where you can be blind to some of the important variation that is actually present in the world. So we've been looking at things like the telomere to telomere reference genome. We've also been looking at the pangenome that's emerging as a way to kind of harmonize and get access to the whole genome in a much more detailed way. So we're very much excited about to do this, although there is sort of a practical challenge of now we have approaching 1 million genomes aligned to GRCH38. It would be quite complicated and expensive to reprocess all of those from scratch, but there's some exciting new algorithms and methods that can do this in a very intelligent way. So one of them, this comes from Ben Langman's lab at Johns Hopkins called Leviosim, where instead of realigning every single read and doing everything from scratch, well, if we pre-compute parts of these genomes that are super similar to each other, we can just very quickly do something called a liftover operation to kind of move it to the new reference genome. And then for the tricky parts on the side, then we can align those from scratch as needed. That makes it much more efficient and also much more cost effective to be able to do this liftover and harmonization. And we're excited about that and a kind of other approaches to do this in a very intelligent and sophisticated way. So we're, in addition to kind of work specifically inside of AnVIL, we're also partnering through that NCPI, that NIH Cloud Platform Interoperability Group, to be able to do some of this remapping, do some of this harmonization. Some of the key data sets like GTAX and some of the cohorts from TopMed, we're going to be processing with these new reference genomes, and those will be available in the coming months and years. And the final thing I wanted to highlight is, AnVIL is an enabler, but I know a lot of people have a lot of questions about the costs associated with it. It isn't running in the commercial cloud. I know that's often the first question that comes up. In fact, that was, I think, your first question last night, was how much does this all cost? Oh, we have some new technology, some new databases and websites to be able to give you some information about that, where we've profiled more than 1,000 tools, and we can give you some estimates about, kind of order of magnitude of costs that are there. Also give you some, guiding principles about how, if you need a more refined estimate, how you can work with that. So I encourage you to talk to Ennis, who's sitting in the back, to learn more about that. Otherwise, we'll have a poster in 2 days at the Biological Data Sciences Conference to talk about this more specifically. So those are a few updates from the Hopkins team. I'm going to pass this off now to Jonathan to talk about some of the updates going on at Broad. Yeah.
We have a question from the virtual participants, and there's one about, can you access all this data?
a lot of the data that we have in AnVIL is a so-called protected access data set. So yes, people can access it, but if it's protected, you'll have to go through an approval process to get to that. That'll be one of the topics that Jonathan's going to talk about in 2 minutes. So maybe we'll circle back to that question after we've gone through that bit of the presentation. Thank you.
Thanks, Mike. Good to see everyone. Glad to be with you all. I'm going to walk through a little bit more of the technical components, particularly in Terra and some of the other products we work on at the Broad Institute that are part of AnVIL, but also just want to help us kind of take a look back at what we've been thinking about in the last year. So a major component of our responsibility at the Broad in this iteration of the AnVIL is to take us to a multi-cloud future. That is something we've been excited about. As Mike mentioned earlier, we've been on Google Cloud for the life of the AnVIL so far, and the next vision has been Azure. We had an original timeline that you see here. Like most software development, we did not follow it exactly. So true to form. And there's some updates here. Some AnVIL team members have seen a slide like this, but just want to call out some updates on this. So originally in July, we decided that we were going to kind of extend the life of GCP-based AnVIL data at least through 2025 through kind of efficient planning on our side, support from NHGRI, and a few other technical improvements. We're pretty certain we can keep the data through 2026, and I think that's just going to continue to extend. We're really optimistic about where things go. So what I'm intending to do here in saying this is to reassure you that our plan is that the AnVIL data will be on GCP into the future, and also as we move to build out the AnVIL functionality on Azure, that'll be on Azure as well. So that's what we're looking forward to. Just as a note, and also a thank you to many of our data submitters. We were a bit concerned when we were going to tell people that, hey, the data is going to stay on GCP. It's not moving exclusively to Azure. And we actually got a resounding positive feedback when we were expecting potentially some disruption. So there was a lot of enthusiasm and support for AnVIL working on GCP. So just good to hear that, and want to share that and echo that. In terms of what's next and kind of where we're at in our own developments, you can think about the future. In summary, we were pursuing a path to build out Azure functionality for the AnVIL using Terra that was very Azure specific. When we took a step back, we said, the future, we really want to be truly multi-cloud, not just GCP and then also Azure, but really to think about what could be possible across other clouds. So we've taken a little bit of a new approach on this, which is slowing us down for now. But our ultimate hope, and not in the slides, but really what number 2 should say, is data is going to be primarily available on GCP. We're moving closely to Azure, and it is our hope, though it's early days, that we'd also be able to offer AWS functionality within the next 4 years. So that's news for most of you, and something we're very excited about. We're still in the early days of that. We hope to have more information on that we will share publicly in the next month. So we're really looking forward to that. I know many folks talked with Benedict quite a bit about exciting data sets available freely on AWS and things that they're doing there. So want to make sure we're serving the community well by going across the different clouds. In terms of a specific component, what we think about, as Mike was talking about, the data is really essential here to data science. So I'm going to zero in a little bit on what we do in terms of the data governance. And if I had to really kind of draw 2 big circles within the infrastructure of the AnVIL, there's really the tools and the data. So just thinking about data here, there's some exciting things we're working on. We are quickly getting faster and faster at the ability to onboard data sets into the AnVIL. That's a big shout of thanks to Kate Balakonis and Nate Calmes on our team, who do a lot of great work on that, and are quickly, kind of even as we speak, getting that process easier and faster for data submitters. So we would hope to be faster, and then also as data sets would come into dbGaP, that they would now kind of come into AnVIL at the same time, as faster, perhaps even before they're available in dbGaP. We're also looking at doing a number of things in Duos, which I will touch on here in a second. The first and kind of biggest announcement is to say is that now the AnVIL data library is fully available through the Duos system. You're able to visit at the URL there if you want to see the specific kind of AnVIL curated library. That means you can now go there, search for different studies and data sets that the AnVIL is hosting through the Terra platform, request access to those. The NHGRI DAC is there. They will respond to those requests. There's a slightly updated process for requesting that data called a library card. That's different than the data use certification you would use in in dbGaP. But once you're through that process, you're off and running and requesting access to all the data sets. And this will continue to be updated live as we release new data sets. So we're very excited about this, 250 data sets. That's the full AnVIL data corpus. Why might you want to think about Duos in the future, and why is it exciting kind of beyond just being another interface for dbGaP? One thing we intend to pilot within the AnVIL is the automated approval of data access requests. So if you're not a fan of waiting for responses from data access committees, what we're looking at is for any data sets that are general research use or health, medical, biomedical use only consented, that those are probably discrete enough that we could automate them with an inference engine we have in Duos and just automatically approve those. We've been talking about this with the NHGRI DAC for a long time and are excited to move in this direction. So just imagine what it would be like to have instant answers to your data access requests to data that's in the cloud. You could be off and running with some of the different analyses we're talking about here in a matter of seconds after you submit your data access request. A thing we're excited about in addition to this with our colleagues at UCSC, we're building out our sample level data explorer that will soon, I think in the next quarter, have an easy way of propagating a data access request when you search across cohorts through that search interface as well. So that will get you over to Duos to make those requests. So we're just excited. Really what we're trying to do is speed up for all of you who are researchers, your time from find to analyze and do everything we can kind of in the middle of the FAIR acronym there faster for you. And as Mike mentioned, just to give you kind of color in the user interfaces here, how are we kind of helping as the AnVIL with the new data management and sharing policy that came out from NIH policy a few years ago? We have Ava and team helped create the DMS helper, which is a way to create a DMS plan that's kind of AnVIL specific. So I'd encourage you to check that out. If you do intend to submit data to the AnVIL, that will do a lot of work for you, probably even better than ChatGPT would. Duos is a place that you could then come and you work with the AnVIL team on that to register your data sets there and then also get them into data storage components of Terra. So we're glad to help you out with that. I know ourselves as grantees and also the NHGRI team, very eager to support researchers using the AnVIL to respond to the DMS policy. With that, I'll hand it over to Robert Carroll.
Hi, everyone. Yeah, I'm super excited to be here. It's great to see everything laid out that we just heard before from Mike and Jonathan. And I think what Jonathan is doing with their team, a lot of it is really providing a platform, right? These opportunities for success. And some of the work that we're doing really has been focused on like clinical and phenotypic data. And so I think this idea we heard earlier, right? AnVIL is that place to share NHGRI data sets. And we have a piece of paper that shows it. But what we really want is everyone to feel that and experience that in a really strong and powerful way. And so getting to that change, there are a couple of boxes I have here that I'll talk about, but I think that's what we're really committed to. And that's something we really want to be able to support, to make sure that it's going to be that place that not the community has to do it, but that they want to do it, and that those tools, again, and support are really there. So there's a couple of things that I think we have very strong right now, one of which is that the sharing of researcher collected data. And that's something Jonathan was just talking about in that question from the chat, right? It is there. The data is shared. You can go access these data sets today through Duos, through dbGaP to get approval, and then actually access technically those resources. And that's awesome. Another product that was, I think, alluded to a little bit is the current findability focused data elements that are populated by the team at the Broad to really help you find and understand. There's a great tool, the AnVIL Data Explorer, that you can go check out that will help you. When you have access, you can drill in and see at a row level some of the data and explore and understand what's there. And so those are some really powerful resources. Some of the things, though, that I think we have an opportunity to improve on and that my team is really focused on is boosting the metadata and provenance that's available, as well as iterating on the harmonized data sets that you can do. And so I think really focusing on some of those metadata repository pieces, and I'll have an example in a second, is so important, especially about things like field descriptions and some of that data set collection provenance, and making sure that's linked and connected to the data that's being shared right now. And then again, I think iterating on that harmonized data that's currently available to really set some expectation on coverage and the transformation provenance. So there's a level of trust in what's there, and you can really intend to use it. And I think this is a great example. This is 1,000 genomes data that's available right now. There's a great interface. You can go look at this data. Anybody can log into Terra, walk in and be able to access this. But one of the things that stood out to me is, what do these fields mean, right? And there is 1,000 genomes documentations out there, but how do we better integrate that with the AnVIL? And that's something that we're really committed to being able to help support. And I think that's something that can help take these elements to the next level. The other side about that harmonized clinical and phenotypic data, I've got sort of an example here, and this is the experience right now to a large extent in AnVIL. There is a set of data that's been pulled across studies, but you end up in these source situations where how is the data articulated? What does it mean in each of these studies? And so we've got 3 sort of simulated example studies here where you've got EHR billing codes perhaps from an eMERGE study and another study, for example, Gregor, where they're using Mondo terms and they're structuring that data in a specific way. And then you've got a third study from RO1 that maybe has a couple of binary indicator variables. Right, for me as a user to answer this question, which participants might have type 2 diabetes, I've got to go, do I read through every data dictionary? Do I scan through all these tables? It's tough to be able to pull that out. And so I think really we want to live in the situation where there's some sense of semantics that's been encoded. There's some sense of data that's there. And this is something that, again, we're firmly committed to providing, some level of quality of service. Now, these things are different, right? Those meanings are different. And I talk about this with my students a lot when we're thinking about how to use EHR data. What are the biases in coding? If you get a billing code for a disease, doesn't mean you have that disease, right? And maybe some of these studies, some of it's going to be participant self-report. Some of it's going to be chart abstraction. Some of it's going to be who knows what. So, you need to be able to go back into the depths. And I think having those references back to the original data when you want to dive in, but this gives you a way to assess at a high level, to kind of understand with some, level of certainty, hey, what information is out there? Who had these kind of reports available? And so I think it's super powerful. And then again, that bottom in the yellow box, we also want to make sure that the metadata that we have, what do these terms mean? How are they encoded? There's a lot of great resources. And by plugging into some of the standards that exist already out especially in those clinical spaces, you can do a lot. You can really empower the data. And I think we can have improved experience for users. The thing is, though, this is a ton of work. Right? To be able to do those links, somebody has to approve it. We want to get to a place with the community where you're actually thinking about this data collection element in a world of sharing, not just for your own research, but to say, hey, it's really important that I use standardized ontologies that describe the data I'm collecting so that when other people want to access it, we've got that clear, well-defined data collected. And this is some work that we've been doing, some ideas, and I copied this. This is some of the technical mapping work that we might use in a study. And you see there's 11,000 rows, right, in this complex JSON. And that's an example of one study. How many different things, what this looks like, how much, really information, knowledge you have to capture in order to be able to do these mappings. And so we really want to make sure that people are thinking about it and data collection. We're excited to plug into that process as data onboarding, provide some standards and some expectations. So I think this is something that we're really committed to, again, expanding that data model, using some of those common ontologies and common data models that exist, and continue to provide some support for that. So again, I think the current findability subset, there's some good information about that that's available now. There's some really strong points to that. Again, we've recently updated how to submit data, what that looks like to submit the data model, some good documentation about what's available. So really interested to hear feedback. We'd love to see people using this. And then again, we're going to continue to iterate on that and make sure that we can meet those needs. Another piece of data submission, something that's going to be launched soon. Again, the goal is to get people to share. Hey, we can plug in using some great PFP import that the Terra team has established. We've got a module that really allows you to do a really cool flow where you can take right from your REDCap. You can pull out those identifiers like REDCap does. You can export directly into your Terra workspace. Now you've got a set of Terra tables. You're one step closer to being able to submit. It's a really great flow, and we'll be launching this soon and make that more available. Right, so we have this vision here where we're going to be able to bring all the data, whether they're from those custom data models that people are bringing in, whether they're using some of the clinical or research standards, to be able to pull that into that core AnVIL data model, bring the data in provenance, and then provide that set of tools once you've got that standard that people can use, supporting interoperability, supporting tools, supporting the use of the clinical and phenotypic data. This is very sort of complicated. I think we're committed. We have a lot of work as a platform that we want to do kind of leading up to that point, where then we really need to get that engagement from the community. And I think a lot of these roles, like we can backfill a little bit, right? But that work to realign data is a lot of work, and we can only do such, some level of good job. We really need the knowledge from those researchers to be able to contribute that back down and make that the standard of data sharing, again, to really reach that standard of fair data. So our goal there, right, we can address these areas, we can work with the community and produce, again, that most valuable resource of the NIHRI, really take stewardship of that data together as submitters and as platform teams to make sure that we've got good, consistent data that's very usable, it's very fair for the rest of the community. So again, we're very excited to continue to push this and drive this forward. So looking forward to working with you all in the future. So I think I'll hand it back to Mike.
Right, so we have a bit of time before a coffee break. So I thought what we could do, if there's any questions or topics that people would like to raise, that would be a great time to do it. I saw there were a few questions online too, so maybe we could circle back to those. Everyone's feeling shy this morning. Questions anyway? Okay, fair.
Yes, I just have a question for Robert about the data model. So, oh, sorry.
This is a question for Robert about the data model because I know it's a continually work in progress type here. If someone wanted to follow, what the current, state is, the current idea is, like, where's the best way for them to get that information?
Yeah, that's great. I'm not sure. I suppose we'll be sharing the slides, but if you go to anvilproject.org, I don't know if I click this link, is it gonna work or am I gonna cause problems? Okay, well, we'll see what happens. Great. So yeah, in the AnVILproject.org, we've got a great set of resources that the team have all generated in this learn section. This step 2, I think, is in the data submission guide here. Yeah, for data submitters. You get down to setting up a data model. There's a lot of good information here that's available. There are references out to the original data dictionary. So there's this sort of more technical details here and sheets that describe each of these entities and what you want to do. There's a good old README here also describing the purpose of this, and then this nice walkthrough here. I think also, and I presume the recording, we recently had a nice chat at one of the AnVIL office hours. I think is available to view too, where I talk a little bit about the model. So if you're interested in more of that conversation, I don't know where to find that. Do you know where?
I believe Steven's uploaded it to the YouTube channel for AnVIL Project.
Yeah.
So it's available there as well as linked out in other places, but the YouTube playlist on it.
Yeah, so check out the AnVIL YouTube channel and you can see again me talking some more about this in detail.
Thank you.
Thanks, Rick.
Follow up on that one. Any thoughts on retroactively applying this data model to the existing data sets, or just curious where that's at? Or
Yeah,
if
that's a
it
great
might be-
question. So a lot of this extraction has been done at a high level pass by Nate at the Broad. And so that is available. And when you go to the Terra data repository, you check out those AnVIL underscore tables. That's a reflection, I believe, of this data model information that's there. I think this is something that we want to launch. Again, I think they've done a great job of grabbing the data available. It's a huge body of work. There's a ton of data that's available in the AnVIL. I think what we're going to do as we go back, we're going to make some iterations on the model, and we're going to, how to say, it's a different mindset that we're going into. Like, my goal is to be able to assure you that, hey, we went through the data sets, we found every information about diseases a participant might have, and we're giving you some level of guarantee that, hey, we've done this. So that's what we're working on now. That's a project we're launching now, where we'll be able to have the provenance there about how the mappings were done. You've seen some of what I showed before, and all of that information, that metadata will be there and available. So that's something that we're launching now.
And if an analyst wanted to jump on that, the Terra data repository would be the kind of API path to get at it.
Yeah, right now, all those data are available in those AnVIL underscore tables within the same place that the data lives currently. That's there in the TDR.
Thanks.
Mhm.
There was a second question from online asking, if we want to teach AnVIL workshop in our university, can you help?
Yes. But I think I'm going to defer, maybe David, you want to talk about this?
Yeah, sure. We'd love to help. So I shared a couple resources on the Slack. The first is our instructor guide. So this is a little bit of how you might set up things on AnVIL to teach a workshop or teach maybe a module in class. And then we also have the AnVIL Champions program that we're hoping to kick off our first cohort soon. And so that's a way to basically liaise between your community and our team, and, you know, feel like you can report to us the needs of your community and basically have a pretty close relationship with us and be able to build out that material. And so there's some resources on AnVIL Champions as well that I shared.
If there's no, oh, private starter question.
Well, I was just gonna have a follow-up, I guess, for Jonathan, because one of the questions about getting into the classroom oftentimes is about getting access to controlled access. And Duo seems like it may sort of help for the instructors that are trying to get there. So for institutions that are not yet using Duos, if you have an instructor, who do they need to contact? Is it just their signing official, and those are the two people that need to get enrolled into using Duos as a mechanism?
Yeah, exactly. The same person, if you're historically used to dbGaP, that would sign up on your dbGaP request from your institutional signing official or authorizing official, there's various names. That would be the person. They'll just need to log into Duos once, click 2 buttons, and they will never need to go back there for you again. So you can commend them that it will be much easier for them than other software where they need to approve every request.